Welcome to the SBGrid YouTube channel. Software tutorials by developers, lectures by structural biologists, unique content brought to you by SBGrid.
Hello, everybody. Welcome to the 2022 SBGrid weekly webinar series. This is our first one of the year. I hope everybody had good holidays, good break. And today we're fortunate to have Jesse Hopkins from BioCAT and Illinois Institute of Technology, who's here to talk to us about BioXTAS RAW. And Jesse's been working with small angle scattering for quite a while now. So with that, I will turn it over to you. And thank you for joining us today and for presenting.
All right, thank you very much for having me. I appreciate it and the opportunity to talk a bit. So yeah, my name is Jesse Hopkins. I'm the Deputy Director at BioCAT, which runs a small angle scattering beamline at Sector 18 at the Advanced Photon Source. And what I'm going to talk about today is BioXTAS RAW, which is an open source program for biological small angle scattering data analysis. So what I'm going to do, I have a couple very brief slides on SAXS so that everybody's hopefully on the same page. Then I'll talk about the program, go through some of the basics of what it can do a quick demo, switch back and talk about SEC-SAXS and how the program can analyze that kind of data, and then do another quick demo of that and finish with a couple more slides at the end. So just to kind of put everybody on the same page, I want to spend just a couple minutes talking about solution small angle scattering. And so the idea here, of course, is we study molecules in solution. And this, in many ways, is very advantageous. You don't need crystals, you don't need to freeze it, you don't need labels. There are very few size restrictions. And so it's really a very versatile technique. And I think that's reflected in the growing popularity of the technique over the last 20 or so years. And so one of the advantages, of course, we have being in solution is we can study a wide range of buffer conditions. You can change the salt, the pH, the ligand concentration in ways that other techniques may not be amenable to. And so this lets you access different parts of the phase diagram for your protein or different aspects. You can study oligomerization as you change the protein concentration, shape change as you add in a ligand. And I think particularly small angle scattering has proved really powerful for studying things like flexibility and disorder, but also complexes and mixtures, things that are challenging to study with more traditional high resolution methods. It's not to say though that SAXS is kind of the be all end all of solution techniques or structural techniques. It's really most powerful when you combine it with things like crystallography, cryo-EM, molecular dynamics, NMR. That's where you tend to get the most interesting experiments and the most powerful use out of your SAXS data. So this is just a slide I put together a couple years ago. I went through the papers from the previous year, 2018, and just pulled out a few examples of things that people are doing with small angle scattering. I'm not going to go through this list in exhaustive detail because there's too much on it and it's not the focus of the talk, but there's a lot of things you can do with small angle scattering, everything from just getting the basic size and solution to validating a high resolution structure matches the solution data, up to doing kind of constrained MD where you're using SAXS as one of the constraints to help refine the structures into a more physically reasonable state. And so there's a lot of different kind of levels of approaching SAXS data and a lot of ways of using it that can be really useful for your different types of experiments and studies. So how do we typically do small angle scattering? We have in very cartoon form, we have an X-ray source, produces a beam of X-rays, or it could be neutrons as well, but I do X-rays, so I'm going to say X-ray all the time. Produces a beam of X-rays, we shine it on the sample, that scatters onto a detector, and we measure that scatter. It turns out that proteins or other macromolecules in solution don't scatter very much more than the solution by itself. So we measure the protein solution, we measure the kind of solution that it's in, the buffer. We subtract the scattering of the buffer from the protein, and we get the scattering of just the protein by itself. And I've shown that schematically here. And I've also shown it in actual measurements down on the bottom. And so usually what we look at for SAXS data is we look at the scattering profile, which is the intensity versus Q vector. Q is just essentially the scattering angle, parameterized in a way that's more useful. And this one-dimensional profile is what contains all of the information about our standard solution SAXS experiments. So that's the very brief 3-minute introduction to SAXS. Now what I'm going to do is I'm going to spend some time talking about RAW and what it can do and how it can analyze SAXS data. So BioXTAS RAW, or RAW as I'm going to call it for the rest of this talk, is a program really built for analyzing biological small-angle solution scattering data, both X-rays and neutrons. And the one historical note I'm going to provide is it was initially developed by Soren Skue as part of the Biological X-ray Total Analysis System project, hence the name, BioXTAS. Over the years, we've expanded RAW quite a bit. These days, kind of the main functionality is summarized in these kind of 5 bullet points. So you can take 2-dimensional detector images into 1D scattering profiles. You can then do basic data reduction on those profiles, like averaging and subtraction, carry out initial analysis for kind of data validation using Guinea fitting and molecular weight calculations, do more advanced analysis, including indirect Fourier transforms and 3D reconstructions, and also some basic and advanced SEC-SAXS data processing, which I'll talk about later in the talk. If you haven't used RAW, but you've otherwise analyzed SAXS data, you've probably used either Primus or Scatter. It's very similar to those in functionality. RAW itself was originally designed to provide a GUI interface for all of this, and so that's where we focused most of our efforts. But in the last year or so, we've also released a Python API, which allows you to create custom processing scripts using all of the RAW functionality. So that's something, again, I'll mention a little bit later, but is kind of an exciting new development for us. So RAW itself is a free open source Python program. And for me, at least, I am a big believer in open source when it comes to scientific data analysis. I like the fact that, at least in theory, all of the analysis can be inspected. You can go into the program, go into the code, and verify what it's doing. Not that I expect anyone ever does that, but I think the ability to do so is very powerful and very important. And I also like the fact that all the algorithms and heuristics that we come up with and we put into the program are freely available to the community. So if someone wants to use it in a way that we haven't really designed it for, or they want to expand on it or refine it, they don't have to start from scratch and try to reverse engineer what they've done. What we've done, they can just take the code and use it directly. RAW itself is reasonably popular, I like to think. Our download site has about 3,000 downloads a year. It's available through SBGrid, of course, which is why I'm giving this talk. We have documentation, which receives a large number of page views and visits per year. And we've also recently created a number of YouTube tutorial videos, which got a good number of views last year, at least as far as kind of scientific software. You know, we're certainly not earning any money on these. We're not influencers at this point. So the documentation is is extremely exhaustive in my mind. All the different things that you can do with software, basically, we have these step-by-step tutorials and tutorial videos, text, images, kind of what you should be looking for, where you should be clicking. So it's really designed for you to be able to learn how to use the program on your own without having to have someone around who's an expert in it. And based on the amount of use the documentation gets, I like to think that it's reasonably successful at that. So that's kind of the overview. What I want to do now is dive into some of the functionality of the program. So I'm going to start with an overview of kind of basic functionality and go through functions in the order you might do if you were analyzing a data set. Then I'll do a brief demo of kind of this portion of the functionality. And then we'll go back to doing some more talking about some more advanced pieces. So if you were to open up RAW, this is what you would see. This is with some data loaded in, but there's kind of 2 main areas I want to draw your attention to. So there's the control panel here and the plot panel here. So the control panel is where you interact with data either on disk or loaded into RAW, and the plot panel, of course, is where you visualize it. So the first step in your data analysis, particularly if you have a home source, if you go to Beamline, this is usually done for you these days, is you're going to collect data as two-dimensional images. So you need to reduce that from these 2D detector images into one-dimensional profiles. And so RAW provides the capability for doing that. You can load in the images, you can carry out Q calibration automatically from various standards, you can mask out the beam stop and detector gaps and anything else that you don't want to have in there. You can normalize the data set by measured counter values like the incident or transmitted intensity. You can carry out absolute calibration. And then once you've done all that and you've set up all these pieces, we then, we can read in a number of different image types. We mostly use the FabIO library for that. And then we can do the radial averaging from these two-dimensional detector images into one-dimensional scattering profiles. And for this, we use the PyPhi library. So this includes a number of different kind of geometries and data corrections. It's also built so that you can outsource the radial averaging onto your GPU, so you can do it quite quickly, which is important as we get these larger, higher pixel density detectors and faster data collection. So once you've done the radial averaging, typically for an experiment in what we call batch mode here at BioCAT, which is where you load the sample and the buffer directly into the cell and make a measurement, you might end up with somewhere between kind of 5 to 20 images for both the sample and the buffer. We take these repeated images to ensure that there's no changes as we measure the data. We usually attribute to radiation damage if we see it. So you'll have these kind of, set of a small number of images that you've turned into scattering profiles. The next thing you'll usually do is you'll check to make sure they're not different from each other, and then you'll average them together. Once you've averaged them, you carry out the background subtraction, you subtract the buffer from the sample. And then, you may do other things like merging in data from multiple detectors, rebinning the data to improve your signal to noise. And these are all kind of basic data reduction processes that RAW can carry out for you. Once you've generated a final subtracted scattering profile, that's when we do kind of standard data analysis and quality checks. So RAW supports Guinea fitting, molecular weight by a number of different methods, checking for differences between profiles, Kratky plots, dimensionless and normalized Kratky plots, all of the kind of usual typical analysis. So, the first thing you might do is a Guinea fit. We do this to look for usually issues with the data. Is there aggregation? Is there repulsion? Is there an issue with the buffer subtraction? All of those can show up here. And so you open up the Guinea window, you'll see essentially this. It automatically tries to find the best range of the fit for you, and then you can manually adjust the fit range and get out the results. Once you're satisfied with this analysis, the next thing we typically do is calculate the molecular weight. So in SAXS molecular weight, typically you don't want to use this to determine the molecular weight of an unknown macromolecule. We usually use it to verify, say, the oligomeric state in solution matches what we expect. So in this case, we were expecting something that had about 172 kilodalton molecular weight. You can see we're getting a range, but it's more or less matching. So it's confirming the oligomeric state that we were looking for. After that, we might take a look at kind of the flexibility or extendedness of the protein, usually using a dimensionless Kratky plot, which again, RAW will do for you. And you can compare different profiles directly on here to get an idea of what kind of shape you have in solution. This is the first real shape information that's coming out of your analysis. And then once we've done this kind of Kratky plot, typically we'll do a P of R calculation. So this calculates the pair distance distribution function. So here we're essentially calculating the number of pairs of electrons at some distance in the macromolecule, and then it's weighted by R squared, which is why we get this going to 0 at 0. RAW supports two different methods for doing this. We have Bayesian indirect Fourier transform method, or BIFT. This is completely automated. It automatically determines the Dmax. automatically determines the smoothing function, the alpha, and then it calculates the P of R function for you. We also, if you have ATSAS installed, support a nice GUI interface to Genome, which is what most people use. So here, if you open the window raw, we'll try to determine the Dmax for you, and then it'll adjust it as necessary. You can adjust it manually as necessary to refine the fit, as well as a few of the other adjustable parameters. Once you're happy with the P of R function, it goes into the main raw window, and you can work with them from there. Typically, the next thing you'll do with the P of R function is you'll do some kind of shape reconstruction. So again, RAW supports two different ways of doing shape reconstructions. The first is kind of the more conventional envelope reconstructions or bead model reconstructions, which does require a separate installation of ATSAS. So we have interfaces to DAMIF and DAMIN, DAMAVR, DAMCLUST, SUPCOM, ambimeter, all these things. So essentially, you just set up all your settings in the GUI, tell it to start, it'll run it all automatically, and then at the end, you can generate this. It generates automatically actually this very nice summary of all the different kind of pieces of the calculation, so you can validate whether or not the reconstruction is good. We support a very similar thing for direct electron density reconstruction from the scattering data. So this is using the DENSE method that was published about 4 years ago now. And here you can actually get real low resolution electron density out of scattering data. So this is kind of showing here nicely where you can see this higher density in the shell and lower density in the core. So again, you just set up all the settings and then you run it and it generates these summaries once it's done. The final thing, of course, in this kind of standard pathway through analysis that you might be doing is generating, saving the results somehow. So you can save all the individual results, of course, to your disk, the profiles, the IFTs, and these all save in formats that are widely compatible with the other analysis programs you might be using. You can also save spreadsheets or really CSVs of your analysis from multiple different data sets. And you can save these automatically generated PDF reports that have kind of nice summary plots and summary tables of one or more data sets that you've done. So I know this is kind of running through things very quickly. The goal here, of course, is just to provide you with an overview. If you really want to learn how to use the program, we have extensive documentation and tutorial videos, as I mentioned. But what I'd like to do now is shift gears a little bit from just showing you slides to actually doing a quick demo of all this functionality. So at this point, you should be able to see RAW on my shared screen. I'm going to navigate in the Files tab to some data that I want to use. So here we have data that we've collected on a protein and its buffer. This happens to be glucose isomerase. And so right now what I have is I have just image files. This is an older data set, so these images are small one-panel detectors, but you can see if I show the data, we have the scattering, the beam stop, it looks more or less like you'd expect. So we have two-dimensional images. That's where we're going to start. So the first thing I'm going to do is I'm going to load in a configuration file, and that's going to show me, or rather load in all of the different parameters I need to normalize out, or rather to turn these two-dimensional images into one-dimensional scattering profiles. So first, actually, I'm going to load the buffer just by selecting it and clicking plot. And you can see that takes these two-dimensional profiles and turns them into these 1D, two-dimensional images, turns them into the 1D profiles we have plotted here on the main plot. I'm going to do the same thing for the protein sample, maybe. There we go. Computer's a little slow today, apparently. And you can see we have a new set of profiles that appear on the plot. If I go to the Profiles tab, I can see all the data that I've loaded in as these reduced one-dimensional scattering profiles. So here, as I mentioned, we typically might measure 5 to 20, in this case, 10 different images of the same sample and the same buffer. So the next thing I'm going to do is I'm going to average them together, which I just do by selecting them and clicking average. And the averages appear on the plot, but they're hard to see because we have these high noise individual profiles. So what I'll do is I'm just going to remove these profiles from the program. They're still on disk. And that leaves us with just these averaged scattering profiles. The next step is usually subtraction. So I mark the one that I'm going to use as the buffer, select the one to subtract the buffer from, click subtract, and I get a subtracted scattering profile. So at this point, we've done all the reduction. We've got a single subtracted profile, and we're ready to do the analysis. So what I'm going to do is I'm going to start, as I said originally, with the Guinea fit. So I can right-click and pull up a menu of all the options, or I can use the kind of most commonly used option buttons down at the bottom. So I right-click, I open the Guinea panel, and what you're seeing now is the automated determination of the best fit range for the Guinea. I think we can probably use a couple more points at the beginning, so I'm going to just tweak this minimum down a little bit, and I'll tweak the maximum down by 1 point as well. So I've refined the range. It's more or less what I expect it to be. You see the Qmin RG and the Qmax RG are good for a globular system. So I can go ahead and say OK. And that information is saved with the scattering profile. And you can see it propagates out into the information panel up here as well. So I'll do the molecular weight next. So again, we automatically open it up. It's going to calculate out the 4 concentration independent methods here. And then if you know the concentration for the measurement, which you should in a batch mode experiment, you can enter that and calculate out the 2 additional concentration dependent methods as well. And these do require calibration of the system. So either for this, it has to be on an absolute scale. For this, you have to have measured a reference of known molecular weight and known concentration. So we do this. This is a glucose isomerase. It's a tetramer. We expect it to be about 172. You can see that for the most part, we're coming in right around there. A little bit of variation high and low, but it's definitely confirming the oligomeric state that we're seeing in solution. Once we've done the molecular weight, typically the next thing we're going to do is we're going to look at the dimensionless Kratky plot. So on this is telling us about the overall shape of the protein in solution. So here I've got something coming up to where these dashed lines are crossing, coming back down around QRG of 4. That's telling me this is very compact and globular. If it were more extended or more flexible, you'd see it coming out higher up here, or having a plateau in this QRG region and coming down later. So in this case, we have something that is more or less pretty spherical in solution and very rigid. Once we've kind of validated all the basic analysis, the Guinea looks good, the molecular weight is giving us what we expect. We have some idea of the overall shape. The next thing we'll typically do, assuming the data looks good, is generate a P of R function using an indirect Fourier transform. So again, RAW supports a couple options for this. Just because it's what people tend to be familiar with, I'm going to use genome. So I select that, it'll open up the genome window, it'll automatically try to find the Dmax. In this case, it does a pretty good job, but if you're not happy with it, you can adjust the maximum dimension manually in whatever range you want to. I can make it much larger, see that it's really not giving us any information past a little bit past 100. So we'll go back to this kind of automated determination. You can also adjust the Q range that you're using. This is particularly important if you're going to do a bead model reconstruction, in which case we need to truncate the Q range we're using for the bead models. So I'm going to do that, and once I click OK, you can see the information from genome is added in the info panel. We've also generated this indirect Fourier transform, or using this indirect Fourier transform, we generated this P of R function that's saved on the IFTs panel. Once we're happy with this, once we've created it and we think it's good to go, the next thing that we'll typically do is do some kind of shape reconstruction. So here I'm going to do a bead model just because it's what most people are familiar with. This generates a lot of files, so I'm going to create a models folder, put it in there. And just for the purposes of keeping this fast, I'm going to do just even 3 reconstructions. I'll go ahead and start that. So this will automatically start each of the reconstructions. It'll run them until they're finished. It'll do the average and give us the results. So this is going to take a little while to run. So while that's going, we can, of course, save the results that were generated. We can save the individual items. just by clicking save. That'll save a .out file, which you can load into most other programs, including all the stuff in the BioXTAS package. It's kind of the standard format for these IFTs from Genome. We can save our averaged and subtracted data sets. And when you save the subtracted data set, all of this analysis is saved with it. So if I were to remove it and then load it back into RAW, it's showing up on the top plot. I can change that. And you can see once I've clicked on it, the info panel has propagated all of this information again. So it does save all that, but you usually want to save this analysis separately somehow. So typically what we'll do, you can either save the analysis info here. This generates a CSV file. Do this for more than one data set that you have loaded in. Or these days, I like to just save a summary report, which I find very convenient. So you can select what items you want to include, save the report. I'm going to call this GI report. It'll take it just a moment to save, and then should have had a Finder window open already. And then once we've done that, we'll have the data set. We'll have the report saved in the folder we selected. I can open it. You can see it's saving this summary plot, summary tables of the results, then more detailed tables for each of the results as well. And so you can have more than one data set in these summary reports, and I find it a very convenient way to kind of both save the results and share them with collaborators who may not be as savvy with SAXS data, may not know how to open up a scattering profile and do the analysis themselves. So at this point, I think it's a good chance to pause for if there are any questions on this basic analysis, and then I'm going to move on to SEC-SAXS analysis. So do we have any questions in the chat from the moderators?
So I don't see any questions in the chat yet. So if people have them, please put them in. One question that did occur to me as you were walking through the demo, though, is there anything in the GUI that would facilitate a user like learning the GUI and then starting to transition to the scripting or the Python API?
There's nothing in the GUI that it explicitly pulls in the scripting. It's kind of a separate. It's the same package, but it's fairly separate. So you usually learn the GUI using the tutorials online, for example, and then there's also tutorials for all the API stuff, but they're fairly disconnected.
Okay, thank you.
Yep. So I'll just continue on. We'll take questions at the end as well, of course. Oh, sorry, flip to the next slide. So the next thing I want to do is I want to talk a bit about SEC-SAXS and then how we can analyze that data set. So this is size exclusion chromatography coupled to SAXS. And the basic idea here is you have a size exclusion system, which I've kind of cartooned out here. You know, HPLC pump or FPLC pump, load your sample into a loop, then pump the sample through this size exclusion column. We take the output of that, the eluate, we run it usually through a UV detector. So this is kind of your standard size exclusion experiment would stop here. But then instead of running this to waste, we actually run it through the X-ray cell. We measure data continuously as the elution happens. And so you end up with kind of the equivalent of a UV chromatogram, but for SAXS, in that you have, you know, UV chromatogram is UV absorbance at every point, sometimes one wavelength, sometimes multiple wavelengths. Here we end up with scattering data at every time point. And so this is extremely useful if you have, say, samples where you have macromolecules of different sizes, because the column will separate out those different sizes, and then you can get good data on each one by itself. That's the idea. Doesn't always work that way. So SAXS, as you may know, is extremely sensitive to having a monodispersed and homogeneous system. So you want, you know, only the protein of interest in there, and you want it all in the same oligomeric state. So size exclusion chromatography, it takes out the aggregates. It automatically provides good buffer matching, so you get a buffer exchange on the column. In the right choice of column and for the right samples, you can usually get at least some separation between oligomeric states, and often between complexes and, say, the subcomponents of the complex that you may have to overload in solution to get the complex to come together. And so it's a really useful technique for kind of extending the range of SAXS in terms of what we can measure. On the other hand, it is a little more challenging to analyze. A typical experiment, instead of yielding 10 or 20 scattering profiles, yields hundreds or thousands of scattering profiles. And so the community in general over the last 10 years has come up with kind of a relatively standard way of doing this analysis. And the first thing we'll do is we'll take each of these profiles, we'll calculate the total intensity in that profile. So basically just sum up all the intensity at all the different Q points. And then we'll plot that versus essentially elution time. And so that we use to determine the interesting regions of the experiment. So nice, flat, low scattering out here, this corresponds to buffer. When protein comes out, it scatters more than buffer, so we see a kind of a peak, and then we come back down to buffer at the end. And if you look carefully, there's a couple small little peaks out here, which in this solution are corresponding to some kind of larger junk, maybe oligomeric states, maybe a little bit of aggregate or something that we've managed to purify away from this main protein peak. And so this lets us kind of determine the useful regions of the data set, and then we'll kind of average some frames in the buffer range as our buffer, average some frames in the peak as our protein, and carry out the analysis from there. There's a little bit more to it than that. So we'll usually pick a buffer range before the peak to use for the buffer subtraction. We'll average it, then we'll subtract it from all the frames in the protein peak. Well, then for each of those frames, we'll carry out an automated analysis where you can calculate radius of gyration and molecular weight across the peak. And what this will tell you essentially the useful range of the peak. So if you look at the data set here on the left, it's nice and flat, this RG across the peak. This is telling me that even though the concentration is changing, because we're going through the solution from the column, which has this characteristic Gaussian-ish distribution in the concentration, what's in the peak itself is not really changing because we have a constant size. If you look on the other side, though, you'll see we have instead this pretty significant change in the radius of gyration on the leading edge, and then it levels out on the trailing edge of the peak. And so this kind of curved RG profile is telling us that we have some other species that's larger in size in this part of the peak. And if you look closely, you can see there is a shoulder here. This happens to be BSA, and we picked a column that doesn't separate the monomer from the higher order oligomers that we know are there. Usually there's a dimer, a trimer, and then maybe a tetramer or something higher order, and all of that's kind of getting lumped into this shoulder here. We have this large change in the radius of gyration. So for this data set, we'd pick something that's over here on the right, where we have this nice flatter range in the RG, and we know that we're avoiding that contaminant, basically. Once we've found the good region of the data, we'll average it, and we'll send it to our, average that, we'll take that subtracted profile, and we'll carry out all the analysis that we usually do. RAW, of course, facilitates all this, which is why I'm telling you about it. You can make these intensity versus frame number plots. You can plot the RG and molecular weight across the peak. You can actually pick the buffer range either in an automated or manual fashion. And then it carries out the subtraction. It automatically generates these RG values, molecular weight values. You can pick the range and the peak that you want to use. And then you can average that and send it to the rest of the program and carry out the analysis I just showed you. So we also have some more advanced features for the series data analysis that I don't really have time to get into, but I do want to mention. So often it's the case that you can't fully purify your sample, right? The monomer and the oligomer maybe are too close together in size to fully separate on the column of interest. Or in some cases, I've even seen small amounts of conversion between the different oligomeric species, between the column and the cell itself, if it's a very rapid conversion. And so RAW supports different ways of kind of handling these challenging data sets. The ones I want to highlight are really the deconvolution approaches. And I'll mention that these were initially developed by the Ando group at Cornell. So what we have is we have two different approaches for deconvolving overlapping peaks in SEC-SAXS data. The first is evolving factor analysis, or EFA. The second is regularized alternating least squares, or regals. Regals is also kind of more general purpose. You can apply it to other types of series data, concentration or titration series, time resolved data sets, things like that. EFA is simpler to use, I would say less touchy, and focused kind of exclusively on SEC-SAXS because of the assumptions it makes. We can also carry out baseline corrections if we happen to have a drifting baseline because the instrument or radiation damage or something like that. And in these ways, we can improve the overall data quality and hopefully get good results out of things that otherwise we wouldn't be able to treat. So what I'm going to do now is I'm going to go back to the program itself, and I'll show you a demo of how to do some of the series analysis. First, though, I'll pull up the reconstructions we were doing. You can see that those have finished. We have this nice summary of what things look like, chi-squareds, RGs, Dmaxes, estimated molecular weight from the reconstructions. We can look at the fits to the data and the normalized residual and get out all the other usual statistics. This is all saved in a CSV file, and these are saved as PDFs, so you can look at them later as well once you close the window. So what we're going to do is we're going to look at series analysis. So I'm going to just navigate to some series data. So here I have 300 something scattering profiles making up a series data set. So I'm just going to load them up, select them up, and click plot series. And then I can go to the series tab in both the control and the plot panels, and you can see I have this integrated intensity curve that I was just talking about, and I've loaded in a series over here. This is actually the BSA data I was showing a couple slides ago. So you can see we have this shoulder and we didn't get this complete separation in our experiments. So typically the first thing we'll do is we'll set the buffer region, we'll calculate out that radius of gyration. So here again, we select it. We can either use the LC analysis or right click and do more options there. And this will open up this liquid chromatography series analysis window. So the first thing we'll do is we'll pick a buffer range. What I'm going to do here is I'm just going to use the auto feature to find an automatic buffer range for this data set. And then if we wanted to, we can manually refine this range. So you can see the range it selects out here in green. And I can expand it or reduce it. I can also just interactively select it on the plot by clicking pick and then clicking the range I want on the plot. I could add an additional region, so I wanted to add one right here at the end. So we'd have multiple buffer regions and remove the regions. But for the purposes of this demo, I'm just going to go with the automated plot here. So I'll set the buffer. That's going to average it. It's going to subtract it from every scattering profile in the data set. And it's going to carry out this automated determination of radius of gyration and molecular weight across the peak. So we're seeing that displayed here. You can see we have this large RG coming down to something that more or less levels out. We can look at the molecular weight by a couple different approaches and see essentially the same thing. You can zoom in on the plot if we want to get kind of better resolution on what ranges we're looking at. It's not so necessary with this data set. And then once we're kind of happy, we can try to pick a sample range. So again, we can try to do that in an automated fashion. Sometimes this works pretty well. For this particular data set, I think it cheats a little too close to this part of the data set because it's trying to capture this strong portion of the peak here. So I'm going to just move it to this kind of flatter region out here. Once we're happy with that, we just say to profiles plot. And when you've selected a data range, either for the buffer or the sample, whenever you try to do something, set the buffer, average the sample range, and send it to your plot, it's going to look through the data set and it's going to say, carry out some automated tests on it and warn you if it sees anything it thinks is suspicious. So here it's telling me there's a couple of things that might be an issue with this data set, and I can decide to either cancel it and pick another range or just continue. And for the purposes of this demo, I'm just going to continue. And that generates this subtracted scattering profile showing up in green. Change the color. In blue here on the plot. And at this point, we can carry out all of the analysis we've previously done Guinea fitting, for example. There's actually a little repulsion in this data set. I won't get into that. Molecular weights, and all of the rest of it. So that's kind of the basic series analysis. But you may have a data set that's not as amenable. This one, for example, has this contaminant, so it can carry out the deconvolution. I'm not going to go through in detail how you do it, but the basic general idea, you pick the range of data that you want to deconvolve, something like that. You figure out how many components are in it using the singular value plot, and go with 2. And pick the range of these components in the data set using these forward and backwards evolving factor analysis plots. And then we refine the selected component ranges based on the fit of the deconvolution to the data set. So here I'm just going to tweak these endpoints and see the chi-squared plot changing. And then we have, 2 individual scattering components corresponding to the ranges shown here. You can see their corresponding concentration profiles. These are normalized to area. And if we're happy with the deconvolution, we can say, okay, done, and that'll save those profiles again back to the main plot here. So that's how we go through the deconvolution. If you really want to learn how to do it, please take a look at the tutorial online. So I have 2 more slides, I believe, and then I'll be done, and we'll have some time for questions at the end if anybody has them. So the final thing I want to mention, but I'm not going to go into in detail, is the raw API. So as I mentioned at the start of this talk, we recently released an API that allows you to use raw as a Python package in other programs. Essentially, you can install it with a pip install and import it just as you would another package. And this is extremely useful if you want to, say, create an automated processing pipeline. It's the backend for the processing pipeline we use here at BioCAT, for example, or just create custom processing scripts or maybe Jupyter notebooks. There's several reasons you might want to do that. The first and kind of most straightforward is you may have a data set that's not really amenable to GUI analysis. For example, here at BioCAT, we do a reasonable amount of time resolved scattering, and I have yet to write a GUI that will really handle that particularly well. So I do all my analysis for that using scripts using the raw API. Alternatively, I've had some groups come to me and say, look, I don't really like using GUIs for the final analysis because I want to have something that's really reproducible. I want to have something where I can point to it and say, here's everything I did in the exact order, in the exact way. And so if you have this API, if you have it in a Jupyter notebook or something, you can always just open up the notebook, rerun it, and say, here's exactly what was done, here's all the parameters, here's what it looks like. The GUI does save all your processing parameters, but they're a little harder to get at, and it's certainly harder to show someone. And again, there's documentation for that on our website. So with that, really, I'm just going to end with a summary. So kind of why might you want to consider using raw? Well, it's free and it's open source. It's Python-based. I think those are very important parameters. It's easy to install and learn and use. I know installation is not such a big deal with SBGrid, but we do have pre-built binaries for Mac, Windows, and Linux. So download it on your own laptop, set it up, run it there, as well as on whatever computer is in the lab. We have extensive documentation available online, including 120-page tutorial and hours of video tutorials. And it's really designed so novice users kind of at our Beamline or other Beamlines can be up and running relatively quickly and at least get the basics of the analysis done. It's powerful. In this talk, I've demoed how you can go all the way from images to 3-dimensional reconstructions, along with a number of kind of powerful features for SEC-SAXS data. You can do it quite quickly for well-behaved data sets. I was able to start generating 3-dimensional reconstructions within 5 minutes, right? It's compatible. The data is saved in kind of standard formats, compatible with other packages like the BioXTAS package. It's well established, right? It's been used by thousands of users for more than 10 years. It's actively used at a number of different Beamlines and home sources, and it's cross-platform. As I mentioned, it runs on Windows, Mac, Linux. So if you're interested, of course, you can find it on the SBGrid website. You can also check out our websites. I have the a link to the main website with the tutorials. That's our download site, and that's our mailing list where you can send messages and get help, report bugs, and get announcements about new versions, and also things like this talk, which I announced on the mailing list. And so with that, I'm going to stop talking and see if we have any questions from the audience.
Thanks, Jesse. That was a very good talk. That was amazing. And I'm also always happy to hear people thinking about getting scientists to read the code that's doing the analysis and, you know, thinking about reproducibility.
Yeah.
And we do have a couple of questions. And so first one from Kumar Ashikar, and I apologize if I'm mispronouncing your name. Is it better to average the image files or sum them? And does RAW have a feature to sum images?
Is it better to average or sum? It depends on what you're doing. The conventional approach is to average at the one-dimensional profile level rather than at the image level, because it's less work. Summing images I typically only use if I'm looking for features in the image itself and I have a relatively weak scattering system. There's no, I think if you work out the math, there's no gain for summing an image versus radially averaging it and then averaging the resulting profiles in terms of signal to noise. But RAW is built around working with just one-dimensional profiles because that's easier from a memory perspective. So there's no built-in way to sum images.
And this might be a naive question, but when you're doing for this experiment, is this one where the detector would be moving back and forth, and if you wanted to do it in image space, you'd have to worry about aligning them beforehand?
For this experiment, there isn't. There can be either multiple detectors or measurements at multiple distances. It depends on the setup at the beamline itself or at the home source. A lot of home sources these days take advantage of multiple different detector distances to get a wide Q range. And so you would then have to worry about how to merge the images from the different distances. You still have to do that with scattering profiles, but it's easier once you convert it to Q space for sure.
Yeah. Always better to work around the problems if you can manage it.
Yeah.
All right. We've got another one from Ken, and it's how do DNA protein complexes look in SAXS?
They look kind of like this little line that goes down. Now, the answer is a lot of people study DNA protein complexes with small angle scattering, and it depends on what you're trying to say about the system and what prior knowledge you have. But you will get scattering from both of the components in solution. If you're doing neutron scattering, so not small angle X-ray scattering, but small angle neutron scattering, there are ways you can contrast out one or the other components and get just part of the complex. And you can do that with nucleic acids and proteins or just with protein-protein complexes. For small angle X-ray scattering, it's not as easy. Typically, what you do is you measure the individual components of the complex, say the nucleic acid and the protein by itself. You measure them together, and then you typically, unless you know otherwise, you make an assumption that the coming together of the complexation doesn't actually change this overall structure significantly on the scale we can measure with SAXS, right? We can't look at individual residue changes, for example. We're not crystallography or cryo-EM or NMR. So you make the assumption that the overall shape doesn't change when they bind, and then you try to use the individual pieces and the overall piece to suss out a picture of how it's all coming together. And there's a lot more to it than that. You can also do contrast matching in SAXS with protein DNA. Lois Pollock's group has done this quite successfully using sucrose, but that gets a little messy. You can actually have the sucrose kind of caramelizing in the X-ray beam. But there's a lot of different tricks for doing these kinds of things. Typically, though, for a complex like that, I'd recommend starting with SAXS. And if you can't see what you want to see, the SAXS at least gives you good preliminary data to expand out to SANS experiments, which are much more competitive for getting beam time.
Thank you. So, another question that's come in. So you'd mentioned compatibility with Primus. Is there a way to extract data to use it for another software for plotting? And the Origin software was the specific example. That's not one I've heard of, so I don't know what formats that uses.
Yeah, sure. So all the individual scattering profiles that we save, let me revert back to my data set here. So all the DAT files that we're saving, for example, this, are just saved as text files. So let me open this in a text editor here. A bunch of stuff, but so we're just saving essentially 3-column texts. These are tab separated, I think, no, space separated. So you'll have, you know, all the comments and kind of metadata are commented. So you can just load this in. This actually specifies number of data points, column header at the bottom. We have a JSON formatted comment with all of the analysis info that you can load up if you want. And so all of the DAT files are saved in these kind of standard text files that you can easily load into Excel, Origin, GraphPad, Igor Pro. I use Python personally, but that's because I do a lot of work in Python. So matplotlib, you know, you can load that in with a single numpy command if you want. The out files are a little bit more annoying because they are the ATSAS format, but again, it's kind of their original specifications. But again, it's all text, and if you do a little bit of work, you can import it into something like Excel. It's all text space separated. You just have these very different column lengths and no comments. So usually people might import this into, say, Excel and then export just the columns of interest and save it into their plotting program or something like that. But yeah, it's all reasonably straightforward to get it. Some of the particular transformation plots as well, I'll mention, like the Guinea plot where you've transformed it into a log of I of Q versus Q squared, and you have the residuals. If you right click on the plot, you can export the data as a CSV directly, so you don't have to do the transformation yourself. So that's available, I think, for the Guinea and the Kraki plots, my memory serves. So there's a few options there.
Great, thank you. From all the options, it sounds like this is a question that you've run into before. Yes.
A lot of people want to know how to plot their data. And you can save the plots directly from RAW, but there you can't adjust the size or adjust the DPI. They're not made for publication. They're just if you want to show it to someone easily. So
remaking figures for publications is definitely a common problem.
Yeah.
All right. I think one other question, and this may be our last one unless more come in. So you mentioned early on that SAXS is most powerful when it's being used in combination with other techniques. What's your impression of SAXS and how it's impacting the AlphaFold 2 and Rosetta Fold improvements with structure prediction?
So that's a very good question. I've worked with several groups at this point that have been using that have AlphaFold but don't have, you know, experimentally measured high resolution structures at their data sets. And so in several of those cases, what we've actually been able to do is use SAXS as a validation technique for the AlphaFold, you know, predicted structure, which then gives them confidence in carrying out their further analysis. So you can basically take the high resolution structure, predict the profile and solution, see if it matches what you measure. And if it does, then you have reasonable confidence in proceeding with AlphaFold. Some people maybe don't need to do that to have confidence in AlphaFold. I'm still on the side of wanting to see some experimental validation. It's also good if you have, you know, regions that are marked as AlphaFold as ambiguous, you know, your orange or red or whatever it's color coding them as, that those are often more flexible regions like hinge domains or something. So again, there you can take this kind of, if you trust the high resolution kind of solid parts of that, you know, the bluish or whatever in the AlphaFold, and you have some hinge regions, you can actually generate sets of structures with different, you know, orientations of the hinge and compare those to the SAXS data and say, yes, this looks like a good explanation for what we're seeing. If it's more open or it's more closed. And so I think in general, having another way of getting a high resolution structure, I mean, we're already doing this kind of thing with MD structures. So it's not, in my mind, it's not very different from the MD structure and approach. It's just whether or not you have significant confidence in the AlphaFold prediction. And the SAXS can help with that. So.
Thank you. Yeah, I haven't heard anything about AlphaFold taking experimental constraints like some of the older, possibly prehistoric predictions things did, and possibly I believe some of the MD approaches use that as well, where you can do steered MD based on the potentials.
Yeah, you can steer MD. There's actually several very nice. There's one open source package for GROMACS, I think, where you can steer the MD based on the SAXS data itself, which is nice. But I don't think AlphaFold at this point takes anything as input. Certainly not SAXS data. Not that I've heard.
Yeah. Well, that's a question to ask them. So.
Yeah.
All right. Well, one last call for questions, and if not, then thank you again for presenting and for all the work you're doing on this. This is very impressive stuff.
All right. Well, thank you very much for having me, and thanks to everybody for their time. I appreciate it.